Assalamualaikum and hi everyone. We are going to present our topic, which is the detection of SARS-CoV-2 by fluorescence microscopy. We're going to cover the principle, the advantages, the method and the limitations of fluorescence microscopy.
Going to start with the principle. So first of all, the specimen that we are going to observe should be labeled with the fluorescent dyes known as fluorophore. The specimen is illuminated with a light of relatively short wavelength, which is either in blue light or UV rays, which is allow them to pass through. The fluorochrome absorbs shorter wavelength rays and emit rays of longer wavelength that pass through emission filter. Then the emission filter block any residual excitation light and pass longer emission wavelength to detector. Lastly, the microscope forms glowing image of the fluorochrome label microbes against dark background. Fluorescence microscopy enables highly specific labeling of target proteins but possesses too low resolution to reveal details of most virus-associated structures. In order to improve this method, super resolution fluorescence microscopy, which is the SRFM, can be used to further discover the interaction between the host and the SARS-CoV-2.
For the method and process, the preparation of the sample, first of all, viro acid cells are grown on glass cover slip which is infected with SARS-CoV-2 and is filled with paraformaldehyde. Then, a nuclei is stained by host before observed under the fluorescence microscope. The virus is identified by looking at the overlapping color signal, and it is specific for SARS-CoV-2 infected cells. Visualization of virus and their specific component, protein and genome, as well as target structure and event in the host cell can also be seen.
Next, the advantages of using the fluorescence microscopy, which is it can provide high sensitivity and specificity due to antibody staining or genetically encoded tags. Second, it also allows visualization viruses and their specific components, which is the protein and also the genomes, as well as the target structure and events occur in host cells. Third, this technique is easy and quick to perform analysis, and it also helps to observe how specimens behave over time.
The last slide, limitation of technique. First, photobleaching can occur through a variety of mechanisms that are still being investigated, while in excited state, fluorophores may be covalently modified, destroying their ability to fluoresce. Next, excitation light might damage live tissue. The producer of fluorescence microscopy may cause damage to the cell under observation due to both light exposure and the fluorophores themselves. Lower wavelength of light used to excite the fluorophores are particularly harmful to cells, destroying DNA directly and creating reactive oxygen species, both of which have an impact on cell survival. Actually, fluorophores or fluorochrome are fluorescence chemical compounds that are capable of absorbing light from a laser and re-emitting the light within a range of wavelength upon excitation. Okay. Another limitation, the fluorophores employed could interface with the metabolic pathway being investigated. For example, GFP is a big protein that may interfere with the movement of target protein. Last, the excited fluorophores may combine with oxygen to produce a free radical that is harmful to the cell.